I want to talk to you about how we use remote and proximal sensing to phenotype, and I'll tell you later why that is so important in sorghum breeding, because there's a gap in phenotyping that we need to close, basically. So just an overview of my talk. I want to tell you first that we have excellent genetic resources that we have been able to develop, but really phenotyping is now holding back the breeding progress, because we also need to match the genotyping with accurate phenotyping. And then I want to tell you the resources we have now built to address this gap. And ultimately I'll also use a case study. As Alex said, I'm working with the Centre for Excellence in Translational Photosynthesis, and I want to use one of the examples, the things we work on for that, to show you how we want to use these phenotyping resources for that topic. So that'll be the talk, but I want to have a upfront claimer. So what I'm presenting here is really the result of a large team of people working on this. There are quite a large range of disciplines involved in the team, and also there's been a few organisations and funds funding this work. I'll acknowledge all these in my last slide, but I just wanted to make clear up front, this is a large team effort. So I just wanted to tell you, we've really had a genotyping revolution. So basically, genotyping has increased, or sequencing has increased exponentially in the last few years. So these is just a number of papers that have been published on sequencing. And you can see, so even just in the last few years, that has really increased incredibly. How many? So we're talking terabases of base pairs that have been sequenced across the disciplines. And that has allowed us in the sorghum breeding team to use these technologies for low-cost genotyping. So we now can use genotyping by sequencing and a few other various techniques, or even just sequencing the whole lines, which then allows us to construct mapping populations. So we can have traditional biparental mapping populations that we now know a lot about, the markers that they have in them, or the genome-specific characteristics. And then we can even sequence some of our breeding populations, or parents of our breeding populations, and then get access to chromosomal areas of interest, or know what's in the chromosomes. We can construct nested association mapping populations, which are also very powerful mapping populations. And then we can also look at diverse sorghum lines and use just general association mapping. So we can sequence some of these lines and then know what's in the chromosomes. So in terms of number, you can see these is what we have sequenced in the sorghum breeding program. And so that gives us like 1,500 lines that we know a lot about in terms of their genome, and 5,000 lines here in the breeding populations, 26 populations with 1,500 lines each that we have genotyped for these NAM populations. And for this diversity panel, we have now 900 lines with detailed genomic information on them. And in terms of data points, we have 250 million genotyping by sequencing data points on the diversity panel, 500 million of those in breeding populations, and 264 polymorphic SNP markers from resequencing. So just to throw some numbers at you, because we'll later now get some more phenotyping numbers that will to match these sort of things. But so incredible amount of data that we have, but because you also need phenotyping information, that has been really lacking. But sorghum is a good model because it has large diversity, and we have access to wild relatives, land races, and modern cultivars, so different levels of domestication that we can access in the hope to get new alleles that we might not have been looking at with the latest breeding lines. And because of that simple, it has a simple genome, so that made the development of these genetic resources easier, but also more effective, because it's easier to learn in a organism that has 10 diploid chromosomes like sorghum rather than wheat that is complicated. So sorghum is a really good model, and what we learn in sorghum we can probably then apply to other crops that are closely related like maize, sugarcane and millet, but even further wheat, rice and other crops as such. So phenotyping, as I said, is now the bottleneck because we do need accurate phenotyping for genetic mapping. So without being able to go out and actually measure a trait in the field, we won't be able to do much with those genetic resources. And most complex traits, I just used yield here as an example, but there are other complex traits out there. They're usually always affected by genotype by environment interactions. So what that means is if you grow the same genotype in different environments, different years, you're going to get a different phenotype because it's all affected by environmental factors that are affecting the growth of that plant in that environment. So that complicates things because now to phenotype effectively we have to grow these breeding plots in multiple environments, probably multiple years. But that's costly, so each individual yield plot costs about $20, the breeders tell me, just to grow it. So that's just putting it in one field, and that's certainly not counting the labour costs for the phenotyping, so the collecting of information on that plot, which at the moment all that is usually done is flowering time, maybe height, tiller number, so the things that are relatively easy that people can collect. But then if we plant those trials in a number of locations, you can see how that is really a costly exercise before we have even started collecting any data. So some other people have called field high throughput phenotyping the new crop breeding frontier because of that reason. So we need a new phenotyping revolution to match the genotyping revolution. And I've chosen Wilhelm Tell here from my background in Switzerland as my revolutionary guy. And so the enabling technology really that are going to help us or have helped us in the past, for the genotyping it really has been this 454. That stands for low cost, not the number stands for that, but it means it's a machine that has been able to massively parallel sequence. It's also called next generation sequencing. So that's really been the enabling technology in making genotyping so fast, high throughput and cost effective. So, and I think to match that in phenotyping is going to be the GPS, or has been the GPS, the enabling technology, because really in phenotyping phenotyping breeding plots, what we need is to know if we want to measure from remote, like proximal or remotely sensing, then we need to really know which is one plot and where's the other plot. So we need to be able to tell where we're measuring on a very small scale. So 3 meters by 2 meter plots or something like that. And also these things are now becoming very lightweight, so we can probably put them on a drone as well. So that's the world's smallest GPS apparently, that's just been developed. So that means now we can phenotype, as I said, remotely. So, and we can put sensors on, for example, a drone. So here you can see that setup we've started out with. It's just a simple drone, and they can carry a camera of normal size, so it's only red, green, blue camera, or then there are specific multi-spectral cameras like a microsense red edge, and I'll talk to you about what that does. You can attach that to this drone easily, or you can attach a thermal camera even. And then we just need mission planner software, so we can tell the drone how to fly over our breeding plots, and you can just take images at a set rate. But we also wanted to develop something that's closer to the crop and has other benefits, like if it's windy, for example, a drone can't fly, things like that, and just by being closer to the crop we're getting more benefits, and we can attach heavier sensors to this platform. So we chose a tractor, and we've just attached a lot of different sensors to this tractor. And we called it GECKO for genetic and environmental characterization through kinetic observation. So what have we put on this tractor? So we've got a hyperspectral line scanner, a nano head wall, it's called, up the top. So that's the specifics for that. It measures in the range of 400 to 1,000 nanometers and has quite a good resolution, so narrow pixels. And then we also have an infrared thermal camera attached to that top arm. We have a LIDAR, light detection and ranging, that stands for. So that gives you an idea of the 3D characteristics of your plots. And yeah, that's what it can measures a pulse infrared laser measures the variable distances to the Earth. That's how it works. And then we have point spectrometers, so they're from Ocean Optics, and they measure in this range of 349 to 1,028 nanometers, and they also have quite a great high resolution. And we have point spectrometers to get the incoming radiation on top of the tractor. We have sonar, which we haven't sort of done much with, but we can probably also look at canopy structures with that. And we have other thermal sensors also attached to the front arm. And then we have the GPS. Like I said, that's a crucial bit of equipment, so we know where we are. And we also collect some ground weather data with a normal weather station so that we have climatic information at the same time. So I just said to you, I'll explain to you what the MicroSense Red Edge camera does, and it's actually a good introduction into reflectance because a lot of those sensors work on what the canopy reflects. So basically, reflectance is the plant absorbs the specific spectra of the light, but the ones that it doesn't want, they get obviously reflected back to us, so that's why we see plants as green. And that can give us a lot of information, especially in the infrared range, on plant health or how much chlorophyll it has, all sorts of characteristics we can collect from this bit of data. And the the MicroSense Red Edge is just a multi-spectral camera, so it picks 5 wavelengths and then gives us a spectrum for those. And from that we can calculate different vegetation indices. A lot of these have already been published, so people have been using them for quite a while in remote sensing. So we can use some of these pre-published indices, and they all give us slightly different information on the canopy or the plant health, the characteristics of leaf compositions and things like that. So they're already ready available. And we, for example, have used this NDVI and have had good correlation with leaf area. So NDVI can tell us how much leaf area is in each plot, which is certainly a very handy measurement for us. And so the MicroSense is a multi-spectral sensor, as I said, just picks 5 wavelengths. But then we have, on the GECKO now, we have a nano head wall and Ocean Optics spectrometers, and they are hyperspectral sensors. So that means that they measure in each wavelength in a certain area from 400 to 1,000 nanometers in our case. You can also get extended ones. But the principle of gathering data is exactly the same, whether you're using a hyperspectral or a multi-spectral sensor. But now we're getting a lot of data from these sensors. As you can imagine, if this sensor senses every wavelength of the spectrum you're going to get for each run it does, you're getting massive amounts of data. So for example, the nano head wall collects 10,750,000 values just per square meter, and our trials are usually around 2 hectares large, so that's a massive amount of data. I use this other example here. So each UAV flight gives us 32 gigabytes of data, and that's equivalent to 38,000 copies of Harry Potter, the Deathly hellos. So just to give you a comparison, I wouldn't want to read these. Yeah, so the next problem is making sense of this big data. So first of all, we need to be able to differentiate between signal and noise. So each trait, what is the best data, what are the best data points to use, what are the best time points to use, or what combination of points, basically trait functions, can we rely on to give us the best information so that we don't have to get lost in the forest of data. And then we also need to get from the raw data to plot-based data, because the breeders want to just, at a click of a button, look at each plot and say it's got an NDVI of that, or it's got heights of an average of that. So we need a well-designed analysis pipeline. And we need to also keep the raw data in a safe place, so that maybe later, if we want to come back to it, we can do further analysis. If more indices come online, we can then go back to the raw data and go and recalculate things. So we don't want to throw the raw data away. We just want to use what's important now, but still safely store for later the rest. So just an example of that, picking our more predictive signals. So in case of Spectra, this is just a spectrum of a canopy across the entire spectrum. So that's what it looks like. But if we pick 40 wavelengths, just 40 selected wavelengths out of that, we get a spectrum that looks like that. So now we can start to differentiate between leaves and heads and senesced leaves and soil much better than before. So just selecting those 40 wavelengths now is giving us more information. And again here, that's just comparing which sensors are probably the best for each trait. So if you look at the pink and the blue lines, that comes from the Ocean 3, which is that spot sensor I was telling you about. Whereas the other lines, they come from the Nano, which is a line scanner. So the Nano just scans a whole line, whereas the other sensors just take a spot reading. And there is a big gap here between the blue and the pink line, and basically telling us that they can't really tell the difference between plants and soil in sparse canopies very well. So in this case, we would be relying more on the Nano line scanner rather than the point sensors for this particular measurement. And to get to this pipeline, so James Watson has designed this pipeline and written all the software for it, and it really is what makes our whole work stand or fall, because like I said, we can't just look at raw spectra, we have to be able to press a button and get a value for each plot. And this is what his pipeline does. So, I don't want to go through it, but I just want to say that there's a lot of steps involved, and it's really been a lot of work to develop this pipeline to get to, in the end, a pixel that we use for our calculations from the raw data value. And it makes our analysis much faster. So with a traditional method, there would have been 15 manual steps from these raw data values to a pixel that we're using, and 8 minutes of computing time. And this software that's called Extractory has got that down to 3 seconds doing that same thing. So, but apart from being faster, our analysis pipeline also has a 24/7 data transfer and backup system, so the data as we get it can be uploaded to the server, and it uses cloud infrastructures for safe storage but also for seamless expansion. So if computing, we can later do some more computing or new analysis, so it's extendable to new indices and techniques as they come along. And I'm just giving you a few examples here of what they have been working on. So basically the traits that we are interested in. So this one is counting plants from the UAV. So what we have been doing obviously is doing a lot of ground truthing. So we've been collecting the data with the remote sensing, but then also went on the ground and counted plants or cut some biomass samples so that we can compare our results. And that's basically what this is, that we get the correlation between what we have counted in the real world versus what they have done with their analysis, automated systems to get from the image to a plant per plot number. The same we can do with just counting heads, so particular parts of the plant, and breeders are interested in that obviously because the heads are the ones that have the grain on, and they gives you information on how many tillers and all that sort of thing. So that's going to be a very handy thing to have, that we can just automatically count heads. And Wei Guo from Tokyo has been developing some software or algorithms to work that out from images, to be able to pick the heads from the rest of the plant and count them. And then we've also looked at canopy temperature and cover from the UAV. So that's a sort of a thermal image we get from that thermal camera when it's attached to the drone. So that can give us a temperature, average temperature of this plot. There's still more that we have to do to be able to really, so first of all we have to only look at the plant parts and be able to exclude the soil temperature, because soil temperatures can be obviously very, very different to the plant temperatures. They're sometimes up to 70 degrees and the plants are at 25 or 30 degrees. So that's a challenge, and there are other challenges that we're still working on to get this working properly. We've also got canopy temperature from the GECKO now. That's still from the UAV. So what we also had to do was just look at specific segments, because the thermal cameras have a tendency to screw things around the edges. So what we come up with is having different slices of the plot that we look at. So the middle two rows are always the ones that we're interested in the first place, but then we also just took a little slice out of the picture to get away from this distortion that the thermal camera has, and then get an average of these. There's about 10 images that are taken for each plot, depending on the speed that you're traveling with. So from those 10 images, we get a density plot, and we've been looking at which percentiles are our best predictors for the actual average canopy plot temperature. So yeah, that's what we're still working on as well. So this is all work in progress. And then similar with height, also work in progress. So we've got this LIDAR, as I said, but at the moment we're only pointing down, so we're only not really getting a proper 3D reconstruction of our canopy. So we have to work out ways of extracting things like height of the plants, or even where the heads are from what we're getting from this looking down approach. We may have to have another LIDAR attached later on at a different angle to really get a 3D reconstruction of the canopy. So this is also work in progress. At the moment, that's a trial to look at the frequency distributions of the point cloud and get the highest points that way, basically. Once we have this worked out, we can obviously get height over time, so that which also gives us an idea of growth rates of the plants, of the plots. So this shows that our different dates and how the plots have increased in height. And that's another dynamic thing that we've been looking at. So senescence is an important trait in sorghum, and the opposite of senescence is stay green. So we have this stay green trait, and the brown line represents a genotype that has got no stay green, so it's a normal senescing genotype, and the blue ones are stay green genotypes. So we just wanted to see whether we can use these different indices to follow the senescence pattern, or whether these plants actually stay green. And you can see that this senescing type crash is much faster here than the other two stay green types. So at the end of the trial, the stay green genotypes have a bit more greenness left. So this is what we wanted to test, whether we can use this to differentiate between those genotypes. So as Alex also said, I also work as part of the Translational Photosynthesis Centre, another big team, which Robert is part of as well, who sits in the audience. And for this team, our job is basically, or our contribution is to screen sorghum for diversity in traits that confer greater photosynthetic capacity at the canopy level. So that's our mission in the larger team. And we want to see whether we can use these phenotyping resources to answer that question. And so what we need to do is look at traits, obviously, that are related to canopy photosynthesis. And there are different, so there are static traits, but also dynamic traits when you think about that. So on the static side, we have traits like leaf area index, the distribution of light through the canopy, radiation use efficiency, leaf angle, size of the largest leaf, light interception, plant height. They're all related to the efficiency of photosynthetic output that the plants have. And then on the dynamic side, we have things like development, phenology, growth rates, and leaf area duration that can give us differences in photosynthetic capacity. So as I said, we need to pick traits that are really going to tell us what we want to know. And in terms of photosynthetic capacity, really one trait we're really interested is, well, obviously canopy growth, but that's made up of radiation use efficiency and intercepted radiation. So if we can get to, with our technology now, with our techniques, to this radiation use efficiency, we've made a lot of headway in terms of being able to pick one plot from another one to tell whether it's more efficient in photosynthetic capacity than the other one. So that's really what we're targeting at the moment. That's what we're working on at the moment, trying to get to this trait. And we already can do a lot remotely to get to this trait. So we have total incident radiation, for example, from the solar radiation weather station, or from the weather station. And we can get leaf area from this cover that we're getting from the GECKO or the UAV. We can get leaf area duration as well from cover over time on these indices. We can get light extinction coefficient by looking at leaf angle and leaf size distribution. At the moment, we still have to do that from the ground, so leaf angle is not an easy thing, but we're hoping that the LIDAR point cloud will also help us with that eventually, that we can determine leaf angle somehow through that. And then radiation use efficiency is made up of biomass and light interception on this side. So biomass, we can also sort of approximate by Tiller number, cover from this NDVI, height and stem diameter. And ultimately we want to correlate indices directly with RUE so that we can figure out which spectra will actually be determinants of RUE. That's what we're working on at the moment. And once we can do that, we'll have a great way of screening our diverse panel for photosynthetic capacity. So then we have to bring it all together now that we have better resources to phenotype. And what that will allow us to do is to go from the phenotype through this mapping, so using what we know about the genomic structure of these traits, or these genotypes, to the genes, so by mapping. And we might be looking at target traits that we phenotype. As I said, we need our high-throughput field phenotyping, and we need natural variation to go this way, to find new genes that are related to traits of interest. But we can also go the other way. We can go from candidate genes that we know are related to photosynthetic capacity, for example, and then look at their sequence variation and then grow them in the field and observe the phenotype. So there's two approaches that we can take now that we can bring all this together. So in conclusion, we have here at UQ, we have developed a high-throughput phenotyping platform to close the gap between genotyping and phenotyping. And by integrating remote UAV and proximal sensing, we benefit from the advantages of both. So as already alluded to, the GECKO, the UAVs, they can't fly when it's windy, but on the other hand, the tractor can't probably go through your field if it's just been raining and everything is wet. So having both platforms gives you a bit more flexibility to respond to current conditions, but we also have different sensors obviously on each and different viewpoints. So whereas the UAV is obviously higher up, and it's much easier to stitch your images. So with the UAV, we have geolocation points in the field, and so that helps us with stitching the images, basically putting the images together to make up the trial again. Whereas on the GECKO, we're much closer, so that is very much harder, which is where our accurate GPS comes in and our calculation of speed. So with the GECKO, the speed is everything. To be able to keep a constant speed that allows us to calculate where we are is the crux in the GECKO. And we also have our data analysis and storage pipeline that ensures that we can scale later on, that we can go back and look at other things that we haven't thought of, other indices, but also so that we can actually leverage off this initial investment of growing one breeding plot that is so expensive. So that way we can collect so much more data on this one plot, and it will probably be the basis of inquiry for a few years down the track. So that, as I said, this work's been funded from a few different sources. So the University of Queensland has provided us not only with salaries, but also with a major infrastructure grant to put this equipment and infrastructure to put this GECKO together. Bill and Melinda Gates Foundation has had some input, Queensland Government, and then the Centre of Excellence for Translational Photosynthesis, as I mentioned. And we'd also like to thank the Grains Research and Development Corporation for the use of germplasm and background IP that we always use in our trials. And as I also said, there's a large team of people, and they all have various disciplines. So we have people who've been in remote sensing for a while, like Andres, and then we've got James, who's been developing this sensing analysis pipeline. We have an engineer, Mark, and with Scott helps us. He's associated to this project. Andrew Borrell, Adrian Hathorn on the bioinformatics side, and then breeders, David and Graham with crop modelling. And then from DAF, we have people like Colleen Hunt, Emma Mace for molecular breeding, Ken, who flies the drones and does a lot of the image analysis. And then the whole breeding team gives us the seed and helps us with the trial management. And then Bob and Suzanne from ANU have been advising us on which genes to look at, which traits to go for. So yeah, it's a large effort by a lot of people. Thank you.